Hello, everyone. I am Ren Sheng from Cyagen. I am glad to have the opportunity to share the development and design of gene therapy drugs with you today. When it comes to gene therapy drugs, it's a hot economic field, but relatively new in terms of drug development. Taking the drug development of AAV as an example, the US FDA has approved 2 AAV drugs. At present, some clinical trials are being carried out in China, but no drug has been approved for marketing. Today I would like to share the following aspects with you. The purpose of drug development in gene therapy, the process of drug development and design, the design of vectors, the choice of the evaluation models, the selection of detection indicators and a marketed drug. Zolgensma. It's a drug designed for the treatment of SMA with AAV expression of SMN1. I will share some of the design aspects of Zolgensma. In drug development, the ultimate goal must be to bring safe and effective outcomes to patients, which is the ideal situation. Then, in addition to considering the safety and effectiveness of gene therapy, it has a specific relationship with the characteristics of the drug. Its long-term effectiveness needs to be considered, because the cost of gene therapy drugs is relatively high compared with other drugs. Most of the targets of gene therapy are chronic diseases or long-term genetic diseases or indications, so long-term efficacy is a very important aspect. According to the current clinical results, as well as some literature reports, there is evidence that the drug effect can last at least a few years to more than 10 years. Whether it can last for decades or more, this remains to be clinically tested. Although our most important goal is to provide safe and effective efficacy for patients, there will also be some phased goals for a particular period of time. For example, researchers in universities or doctors in hospitals may publish articles after they conduct research. Some startups may be more focused on achieving a milestone in the short term. If you're going to develop a drug, whether you're going to make something that's in the initial stage and whose efficacy is not quite certain, like a medicinal exploration, or for the efficacy has a certain grasp, ready to declare clinical. There is a difference between the development and design of the two. There is also an informal registration for drug declaration. For example, lab researchers can initiate an investigator-initiated trial. There are also registered investigational new drug applications, and the final declaration of listing after clinical phase 123. There may also be some differences in scheme design. Based on the purpose of different stages, there are certain differences in the detection indicators and evaluation models of drug development, which requires specific consideration. For measuring a drug, two indicators are ultimately measured: safety and efficacy. Sometimes for drug optimization, the adjustment direction of safety and efficacy may be the same, and sometimes there may be some differences. For example, if the safety is reduced after the dose is increased, but the effectiveness is increased at the same time, what do you think about that? Different characters may have different priorities. As a pharmaceutical company, whether it is safety problems or insufficient effectiveness, it is undoubtedly a big problem, and they want to improve both simultaneously. But from a regulatory point of view, the safety of the drug is more important, because there is no direct link between drug effectiveness and regulators. If it is a clinician who declares an investigator-initiated trial, he may also consider the safety of the drug. However, as a patient, if he is suffering from a serious life threat, he may have a higher demand for the effectiveness of the drug. If there is no immediate threat to life or other drugs are available, patients may be more concerned about the safety of customized therapeutic drugs. This is a flowchart of drug development for gene therapy, which is actually similar to conventional small molecules or antibody drugs. In the basic research stage, if the in vitro test results in animal models show that it is safe and effective, it will enter the stage of subsequent process development and safety evaluation. If all okay, you may apply for clinical application. Then it goes through phase 123. If it goes well, eventually it will be on the market. The whole process of gene therapy is similar. Of course, as a pharmaceutical company of Party A, it can choose to carry out the whole process within the company. It can also consider entrusting companies like Cyagen as Party B to conduct the corresponding R&D. Currently, Cyagen provides CRO services in the early stages of drug development. Other companies such as CDMO, which focuses on process development, CRO, which specializes in safety assessment, and CRO, which focuses on clinical research, also have some. As I see it, the early R&D service can be divided into the following links: project research and program design, and molecular experiments, virus experiments, cell experiments, and in vivo studies, to preliminarily determine that the drug is safe and effective. Take AAV as an example, it can be divided into the following steps. At the beginning is to identify the corresponding disease, target genes, pathogenesis, cell model, animal model. as well as administration method, AAV serotype selection, promoter selection, and codon optimization sequence screening. Of course, we also need to consider the patent and production issues, and finally give a comprehensive experimental scheme. We also have to think about the overall turnaround and cost. If it is divided into various links, the molecular part may mainly focus on some researches on plasmids, including the design, construction and extraction of vectors, and the preliminary production analysis, preservation and arrangement. The virus section is mainly about the packaging of AAV, including transfection, collection, purification, detection, arrangement, as well as some preliminary research. This part of the cell may be related to the construction of cell model, gene mRNA and protein expression, subcellular localization, gene function analysis, and in vitro efficacy evaluation. Then, the body part is also similar, including the construction of animal model, protein expression, in vivo efficacy evaluation, as well as preliminary safety evaluation, as well as this pharmacokinetic analysis. Why is it necessary to consider the process of plasmid and the initial process of virus in the basic research stage? Because sometimes special situations arise. For example, our vectors may be effective in the design process, have certain therapeutic effects. However, it may have obvious defects in production, like the plasmid stability may affect the virus production and air diameter ratio, which is a significant limitations. And if such a candidate drug enters the production stage, there may be a relatively large risk, causing some difficulties for suppliers. Doing some research and analysis ahead of time can greatly reduce the risk of failure. In addition, the reason why it is said to be preliminary safety evaluation here is that only the virus used in the formal safety evaluation is the clinical disease virus, and it is conducted on large laboratory animals in the phase 2, so in the early stages, it's called a preliminary safety evaluation. Find out this big security problem in advance. This is the whole process of gene therapy drug development, and then share with you some experiences in vector design. AAV is an icosahedral structure consisting of a single-stranded genome and a capsid protein, similar to a sphere. And then in the middle of the virus is the genomic DNA of the virus. Recombinant viral vectors used for pharmaceuticals, which replaces the representative gene and cap gene of the virus itself. With our target gene sequence, including promoter, the CD sequence of the target gene and the terminator sequence. The overall vector design takes into account both the selection of the serotype of the capsid protein and the genome. In the case of serotypes, people now basically know the characteristics of various serotypes. There are some studies on the basic characteristics of different serotypes and their infection efficiency. But back in the early days, maybe not so clear about the research. In 2006, Professor Zhao Guangping made a brand new comparison of AAV 2/5, 7/8 on monkeys. There are some differences in the expression of this gene in the liver. Now, whether it is intravenous injection or local administration, there are significant differences in expression efficiency among different serotypes. When designing a program, we need to fully consider about it. In addition to serotype, the codon-optimized sequence of the target gene also has a great influence on the expression of the gene. The example here is factor VIII for the treatment of hematological diseases, if it is a full-length sequence expressing wild-type or codon optimization, or the codon-optimized sequence of the truncate, its expression level is also several times different. Another thing to consider is the selection of promoters. The example given here is cone-specific promoter of ophthalmology. It is specifically expressed in human retinal cells, monkey retinal cells and mouse retinal cells, which is similar to the cone cells. But the researchers also made a comprehensive study comparing the gene expression of retinal cell-specific promoters between mice and humans, monkeys and humans, and found that the correlation was not very high. The correlation between mice and monkeys was just 0.3, and between mice and humans less than that. The correlation between monkeys and humans was relatively high, which can reach over 0.6. Therefore, when selecting promoters, especially specific promoters, we must fully consider the differences in expression efficiency among different species. Relatively speaking, the similarity of universal promoter expression among different species is relatively high. To sum up, we mainly consider 3 points in the design of viral vector: serotype, promoter and codon optimization sequence. Serotype determines the specificity and infection efficiency of virus tissues and cells, and is also related to immune response. There may be some differences in immune response among different serotypes. Another is the production of the virus. There are some differences among different serotypes and different species. Promoters come in a variety of sizes, some long and some short. The packaging limit of AAV is basically only about 4.7 kilobits to 4.8 kilobits, so large and long promoters have impact on the selection of target genes and other regulatory elements, which needs to be considered. Moreover, the specificity, intensity and persistence of gene expression are also strongly related to promoters. In particular, there are large species differences in the specific promoters. In addition, codon-optimized sequences can affect gene expression efficiency, and sometimes may cause a risk to sequence stability. Especially, sequences that are uncommon non-native codon-optimized sequences are sometimes unstable. It is possible to produce a strong immune response. If the gene fragment is too large and exceeds the AAV packaging limit, we can choose mini-gene or truncated mutants. In addition, if you are developing a drug, you need to take the patent issue into consideration. Sometimes a lot of it is very appropriate with a good serotype, promoter or codon-optimized sequence. But if someone else has patented it, it is necessary to circumvent the patent problem and make some adjustments. Its condition varies greatly because of different diseases and targets. In theory, there is a set of exact rules that apply to all kinds of drug development, but basically, the study can only be done according to specific indications and specific targets case by case. Try to find the most appropriate design for a particular disease and version, and try to have a traceable or verifiable experimental basis. In addition to how to design the vectors, we also need to think about its safety and effectiveness. Most of the time, it has to be judged by some models, because after all, it is impossible to carry out clinical verification directly from the beginning. There are two main types of models. One is in vitro models, such as human cell lines, animal cell lines, and cell lines associated with pathogenic cells, as well as primary cells from patients, iPSC differentiated cells, quasi-organs, etc. There are also many kinds of in vivo models, rodent animals like rats and mice, rabbits, pigs, dogs, monkeys, and various humanized animals. The most common model among them is humanized mice. There are great differences in the evaluation among them. When choosing a model, we should fully evaluate species differences and disease associations according to the purpose of our study. Let me give you two common examples. If we study the nervous system, in vivo models are often pigs or monkeys. If the ophthalmic non-genetic model is used, rabbits or monkeys are preferred. We also need to consider the budget of money and time, the technical difficulty, the mechanism of drug action, the accessibility of models, and the reliability of results. For example, zebrafish and fruit flies are also two common model animals. But if they are used to evaluate gene therapy, it is likely that the clinical relevance will be very low. Just to show you a schematic, some models of retinal-associated AAV drug evaluation. In ophthalmology, such as in vitro models, including the simplest 293 cells or HeLa cells, of course, they can be used, but the clinical relevance is very low. By contrast, RPE cells are better. Then it is more ideal to have human-derived primary cells, or HiPSC differentiated RPE cells. If there are organoids, more multidimensional information can be obtained, which is certainly more appropriate. If it is an animal model, it is commonly used in rats and mice, but the eyes of rodents are quite different from those of humans. Among the large animals, pigs, dogs or monkeys are more ideal models. But if you want to use drugs related to human sequences, such as CRISPR, sgRNA or ASO, humanized animals are preferred because they allow for adequate evaluation of the efficiency of gene editing, gene regulation, or gene degradation in vivo. In general, the higher the clinical relevance of the model, the higher the time and cost required for the development and research of the model. In addition to drug design and models, we should also pay attention to the test indicators, which are often related to the research objectives. If we're doing this in the early stages, we don't have to look at all the indicators. We just need to focus on some of the key indicators that are relevant of efficacy. Well, if you have confidence in this drug and want to declare it, surely it would be better to get more comprehensive data as much as possible. The richer and better the data, the more likely you are to get a clinical approval. In addition, regarding the declaration, we can roughly divide it into two key stages. One is to make an IND declaration, which requires efficacy assay. And then, as much as possible, directly reflect the physiological effects and effects of the drug through the data. However, if the clinical trial is going well and declaring for marketing, we may have a critical potency assay at the product release stage, which may have a different focus. It may pay more attention to quantification, requiring stability and reliability, a direct correlation with the activity and dose of the drug, because it is used to determine various characteristics of your product. If it's easy to quantify and stable, it will be easier for products to be approved. If it is difficult to quantify, or if the indicators is unstable, then you may encounter some problems. The above is about vector design, model selection and evaluation, indicator evaluation. Next, I will share with you about this drug that has already been on the market, Zolgensma, about its design and consideration, as well as the analysis of research properties. The marketed drug Zolgensma, whose indication is SMA, targets patients with SMN1 biallelic mutations under the age of 2. There is also a reason for choosing patients under the age of 2. It is most likely because AAV infection in children under the age of 2 is relatively less affected by the immune response. Now let me introduce the SMN1 gene to you. SMN1 is one of the genes that encode SMN protein. If SMN1 gene is mutated, it will lead to underexpression of SMN protein. It can affect the survival and maintenance of motor neurons, resulting in muscle degeneration and necrosis in patients. As the disease continues to progress, it may eventually lead to death. The SMN1 gene is widely expressed, but mainly in the spinal cord. In addition to SMN1, there is another allele called SMN2, which can also express SMN protein. Different people have different copies of SMN2. Its high expression relieves some symptoms caused by SMN1. SMN1 is on chromosome 5, the CD's length is 885 BP, and the protein is 294 amino acids. This drug uses double-stranded AAV9 with a CB promoter to drive the expression of SMN1. Its clinical administration is a single-dose, intravenous injection, at a dose of 1.1 times 10 to the 14 Vg per kilogram. Well, here we can find some interesting information. Why choose the CB promoter? CB promoter is a universal promoter and the hybrid CMV enhancer. It is probably because it is highly correlated with the expression of SMN genes, and it is widely expressed. A universal promoter is selected to match its expression relatively, and its CDs is relatively short, only more than 800 BP, plus the length of the CB promoter, plus the length of a terminator, may be less than 0.4 K. So you can choose to express it with double-stranded AAV. In addition, the dose is 1.1 times 10 to the 14 Vg per kilogram, which is relatively high. It is administered intravenously, but its target is at least to reach the spinal cord, which penetrates the blood-brain barrier. Although AAV9 has a certain ability to penetrate the blood-brain barrier, it still requires a higher dose. This is the condition of SMN's disease. There are two genes called SMN1 and SMN2 in the human body. Normally, there is a base difference between SMN2 and SMN1, which causes exon 7 to skip when the gene is expressed. Most of the product is a degraded, non-functional protein. A small amount of SMN2's products are expressed as functional proteins. If the patient has mutations in both alleles of SMN1 and cannot express the protein, the SMN protein in the patient's whole body will be significantly reduced, which leads to the atrophy of motor neuron cells. It is well known that people who work out tend to be relatively muscular, while those who exercise less tend to be the opposite. But if necrosis occurs to motor neuron cells, then they can't send nerve impulses to muscles, which is equivalent to the muscle being in a state of continuous dormancy. Finally, muscular dystrophy is a very bad condition for patients. Why choose AAV9 over something else? It may have something to do with its characteristics. This is the applicability of some wild-type AAV serotypes to spinal cord and nerve cells. Relatively speaking, AAV9 has certain neurological applicability and can effectively infect neurons and glial cells. If injected intramuscularly, it has the function of retrograde transport from the muscle to the motor neurons of the spinal cord. If the drug is administered intravenously, AAV9 has the ability to penetrate the blood-brain barrier and can infect the spinal cord cells. This has been proven in newborn mice, adult mice, cats and monkeys, which may be the reason for choosing AAV9. In addition, I notice something special: it chooses double-stranded AAV. In fact, most of them are single-stranded, so why does it choose double-stranded ones? Whether single-stranded or double-stranded, AAV infects the cell and releases the genome into the nucleus. If it is a single-stranded AAV, it needs to go through a step called second-strand synthesis, and then integrate the long-standing appendages, or a small amount of AAV, into the genome, to express the target gene over time. If it's a single-stranded AAV by itself, once it gets into the nucleus, it does not require the step of second-strand synthesis, but instead forms add-ons directly, or is integrated into the genome. In contrast, the expression of single-stranded AAV is faster, with a higher quality. Of course, there may be some differences in production and quality research. Its disadvantage is that its packing load is basically halved, like a rope folding in half. The expression efficiency of double-stranded AAV and single-stranded AAV-SMN has been studied. Figure C shows the therapeutic effect. The yellow column is the expression of double-stranded AAV, and the gray one shows that of single-stranded AAV. In Figure D, the green one indicates double-stranded AAV and the blue-purple one single-stranded AAV. It can be seen from both figures that the expression of double-stranded AAV is significantly higher than that of single-stranded AAV. Then, from the perspective of therapeutic effect, no matter the survival rate, body weight and body size of mice, double-strand AAV has obvious advantages over single-strand AAV. And here you see the expression of SMN in histology, and its effect on muscle. And you also see that the expression of double AAV is stronger than that of single AAV. Double-stranded AAV is also better than single-stranded AAV in maintaining the number of motor neurons and muscle integrity. In this paper, the pharmacodynamic detection of AAV-SMN1 is studied in detail. Different doses were used to see the expression level of SMN, the number of motor neurons, and the size of muscle fibers in the mice. You can see that as the virus increases, its gene expression and efficacy are both increasing. This was directly determined by fluorescence staining in neurons, and the expression of SMN increased with the use of different doses. Here's the functional test, and we can see that the grip, the anteroposteric reflex, body weight, and survival of the treated mice also increased with the dose. When the dose was increased from 1E9 to 5E10, the mouse's index data also increased accordingly. Because the data presented above are all obtained through experiments on small animals like mouse models, there must be a big difference when the drug is really used in humans. Large animals are often used for research, but it is difficult to knock out or other genetic modifications with large animals, so here we use GFP instead of SMN. By detecting the GFPs and expression of SMN in large animals by detecting the expression of GFP, these data were obtained through experiments on pig models, in addition to the corresponding studies on monkeys. GFP is also used instead of SMN to conduct testing. It is important to note that when conducting AAV-related studies in monkeys, the presence of high titers of AAV-neutralizing antibodies must be fully considered, and if so, such monkeys are often excluded because high titers of AAV-neutralizing antibodies can have abnormal effects on the expression of AAV, the efficacy and safety of the drug. Well, now comes the promotion time. Cyagen can provide a one-stop solution for gene therapy, from upstream target prediction, midstream viral vector construction and evaluation model construction, to downstream phenotype comparative analysis, all of which are within the scope of our services. Here is a case predicted by our AI disease model. The patient has a mutation in the COL4A5 gene, which normally causes a change in an amino acid that is not identified in the disease database. Based on the protein structure, it should not cause significant pathogenicity, but in fact this patient is more serious. We use the AI model to predict the consequences of this point mutation, and it can affect the splicing of exon 21 of the gene, resulting in the loss of exons about the length of 36 BP after exon 21. In addition, we made a stabilizing organelle, constructed point mutations on the cell model. Then the fragment was amplified, and it was found that the amplified fragment became smaller. At the same time, mini-gene verification is carried out. The mutant and wild-type plasmids were transferred into the cells, and then the fragment size was detected, and it was found that it was also significantly reduced. In addition, we did Sanger sequencing, and the results were exactly the same as we predicted. 36 bases were lost. Thus it can be seen that the prediction of this AI model is relatively reliable. In addition, we are also optimizing and transforming AAV. We can use AI to assist us in the study of directed evolution of AAV. The approximate step is to synthesize a large-scale library first, and then package it to get the virus library. Measure its characteristics, including the titer of the virus, and its infection efficiency in the tissue. A corresponding relationship is established among the violent characteristics, titer and infection efficiency of the virus. Then let the machine learn and get an AI model. To put it simply, between the specific sequence, like the sequence of amino acids and the characteristics of the virus, there may be some features, akin to an invisible mountain peak. We put markers on the measured features, kind of like dropping seeds on a mountain, distributing them in different places, and then using those markers to outline the whole mountain. If the outline is fairly accurate, we can predict the characteristics of those low and high points and pick out the sequence of desirable features that we want, and then do a specific test. If the prediction result of the AI model is consistent with our thoughts, it shows that the model is quite reliable. If not consistent, we will adjust and optimize the AI model according to the experimental data, and iterate constantly. This model can accurately predict the characteristics of the virus, and its method, speed and general effect are similar to those of directed evolution. Its advantage is that it can greatly reduce the cost and time of screening. Here is a case we did before. We predicted some high-yield and low-yield virus sequences, then packed the virus individually to verify the titer results of virus yield. Of the 12 predicted high-yield sequences, about 8 were significantly higher than those of low-yield sequences. At the same time, among these high-yield sequences, the yield of some sequences was significantly higher than that of wild-type sequences. If the model is reliable, it can effectively predict the yield of the virus. Thus, we can find some better vectors than wild-type sequences as a potential and more excellent drug delivery vector. In addition to the prediction of yield, we also predicted liver targeting. The results shown that these two candidate vectors' liver targeting ability was better than that of the wild type. The actual experimental results also showed that their liver expression efficiency at day 8 was significantly higher than that of the wild type. That's all for today. Any questions?